have any like questions for you or anything.
There you go.
Because you did this last time, right?
Yeah. It says I'm custom live streaming service.
Yeah, so I clicked it and it said setting up your webinar, redirecting to the live streaming page. So now I'm on the YouTube.
Yeah, and Rich said that it won't show up until 9 a.m.
So right now, I'm just on like the YouTube MBO Woods Hole homepage.
Yeah, and you'll see at 9 a.m. there will be another little window that shows the seminar.
Okay, so it should be good. I haven't had to input any of those, like that information that Richard sent us yet.
Interesting. Okay. If they didn't ask you, maybe it saved it in some way. Maybe it's already associated with this.
Ready to start welcoming people?
Yeah. Let's get them in.
Okay.
Have you tried to share your screen yet, Hopi?
Nope. Should I do that?
Might as well.
Should be working.
It works.
Should I share my desktop or my PowerPoint?
Whatever you want to do.
Okay.
Sure you want. Yeah.
Okay. I've never had that option before.
Yeah, it worked.
Yep. We see your screen.
Good.
Fantastic.
Ernan, do you see the live stream?
Not yet. Let me see.
I'm still on that page and nothing has changed.
Morning, everybody. We'll wait maybe another minute or two before we get started. Thanks for joining us.
Yeah, Molly, if you go to videos, it shows up. And I think in a minute it will show up on the main page. So it's working.
Okay, perfect. Thank you.
Oops.
All right, we'll wait one more minute and then we'll get started. Today's talk. Thanks to everyone for joining. All right, I think we'll get started. So good morning, everybody, and welcome to today's lecture for Physical Biology of the Cell, 2020, limited edition. It's kind of a bummer. Today would have been a really, really fun day for all of us to be in Woods Hole, to be followed by lunch at Quick's Hole, but it's not to be this year. Let's hope for next. So it's my great pleasure to introduce today's speaker, who's Professor Hopi Hoekstra from Harvard University. As usual, I'll start out with a few of the biographical facts, and then I'll turn to the more interesting features, at least more interesting to me. So Hopi went to undergrad at UC Berkeley, where she, if I remember correctly, majored in biology. And then she went to the University of Washington for graduate school, got her PhD there, and then was a postdoc at the University of Arizona. And that's actually when I first encountered her in a paper, if I remember correctly, that was in PNAS that really caught my eye because it had to do with coat color of mice that lived in different habitats. And this is actually part, maybe she'll tell us about it today, but maybe not, part of a long history, a very interesting history of people examining field mice for all sorts of different interesting evolutionary and ecological questions. After that, she went to UCSD for about 3 years as a professor, and then moved to Harvard, where she's been ever since. She's an investigator of the Howard Hughes Medical Institute and a member of the National Academy, and et cetera. You can look her CV up and find out for yourself that she's a very distinguished person. I actually just would say that that having taught evolution quite a few times and led many field trips to the Galapagos and New Zealand and Indonesia, often my favorite part of teaching my course, and actually I hope he knows this very well, I just got a hard copy of a new book that I wrote, which I wouldn't normally say, but I'm saying it because I consecrated a whole section of that book to Hopi and her work. And And I guess the thing that I think is really neat in her work is that it combines in a very beautiful way field work, like in the most interesting way, whether it has to do with coat color, or I don't know if I'm going to use the right words, but the holes that are dug. There's a super interesting story of she and her grad students, I think in like Nebraska or something, North Dakota, I can't remember, but super fascinating. And so I love that. And we were just talking about our new online era. And actually, I personally have participated in online lectures for a long time. And I was just thinking yesterday on my way back from the mountains that the two people I have listened to the most on bike rides are Richard Feynman and Hopi Hoekstra, which is really pretty funny. So you should go grab her iBio seminars. They're really something. In general, if Hopi talks, she's worth listening to. Anyway, I probably embarrassed you a little, so it's a good time for me to exercise mute, which I will do. And as always, thanks for joining. And let's get your, as we always do, let's get your date for next year on the calendar today. And hopefully we'll have lunch at Quixhole next year. Thanks again for coming.
Great. Thanks, Rob. It's hard to live up to that introduction, so I'm all flustered now. As usual, you give me way too much credit, and I'm blushing. So let me take a deep breath and start by saying it's always a pleasure to visit this group, although this year, of course, virtually. I encourage folks to ask questions during the talk. I purposely tried to make this talk shorter than usual so that as questions come up, please interrupt. And I guess through the question and answer panel, and I'm happy to address questions. So today I sort of have a slightly ambitious goal, in part because I'd like to tell you about a relatively new story that's emerging from my lab group, in which we try to connect evolution, genetics, development, and behavior. So let me start. Let's see. Okay, I'll do it down here. By the big question that motivates the work in our lab, and that is, we're really interested in understanding how organisms adapt to novel environments and adapt at multiple levels. And this sort of comes in two contexts. One, we're interested in animals that move to novel environments. So for example, the colonization of islands. And so all of a sudden, organisms are faced with an entirely new ecosystem. But we're also interested in when animals stay put and the environments around them change. And this is certainly becoming increasingly important in the modern world. So how do we go about addressing this question? There's lots of approaches. The way we've been addressing this question is to start at the level of trait variation. So picking a trait that we think is important for adaptation and trying to dissect it genetically. And the reason we do that in part is because once we understand something about the genetic variation that gives rise to these differences in traits, we can use those genes as sort of a handle to address two important questions. And that is, what are the evolutionary processes or the ultimate mechanisms driving variation in those traits? As well as the proximate mechanisms. What are the changes in the molecular or developmental mechanisms that give rise to this trait variation? So in other words, by addressing these questions, we really get at both why traits vary and how they vary. And so that's the framework in which we're gonna talk today. So I would say over the last several decades, there's been an increasing number of cases in which we've been able to sort of achieve that goal, primarily having to do with traits that are morphological traits and understanding how genes act through developmental pathways to change morphology and how that variation in morphology ultimately affects an organism's ability to survive, to reproduce. And as Rob mentioned in this introduction, his embarrassing introduction, we've spent a lot of time working on pigment. And when I first started my lab in UC San Diego, we decided to work on pigment in part because it had all of these very convenient traits. One, it was a trait that we could measure quite reliably. We could measure hair color either on the whole body or on individual hairs. We had some hints about why color might vary in the natural world, so some sense of how it could affect fitness. And also we could rely on over a century's worth of work by mouse geneticists who in laboratory colonies had identified genes known to have pigment effects, so these pigmentation mutants. And also some understanding of how those genes act through development. So this was almost the perfect scenario for trying to make these connections. And sort of emboldened by the success that we've had with pigmentation and other morphological traits, we sort of decided or asked the question, could we apply the same approaches for making these connections for morphological variation to behavioral variation? Now this comes with its own set of challenges. So for example, behavior is much harder to measure, for example, than morphology. We know much less about candidate genes that might be involved in behavior and how those act in or on neural circuits, the brain being one of the most heterogeneous complex organs in the body. And it may be harder to understand how behavior can affect fitness in the wild, in part because there's also often a learned component as well. Nonetheless, those challenges didn't stop us, and we've started to work on trying to make these connections, our lab and other labs. And recently we published a paper identifying a gene that affects parental care in our favorite group of mice. But we've decided that we could try to be even more ambitious than this, because if we think about when organisms either move to new environments or the environments around them change and they adapt to those novel environments, it's often not just morphology that changes or just behavior, it's both of them that change in concert. And so what we really want to know is how genes affect both morphology and behavior and how that interplay between morphology and behavior affects fitness. So this is the long-term goal, is to understand all of these connections to get a much deeper understanding of how organisms adapt to their novel environments. Okay, so with this context in mind, let me introduce you to the system in which we're tackling this challenge. So this is the deer mouse, Peromyscus maniculatus. It's the most abundant mammal in North America, and I would argue the cutest mammal as well. We refer to them as charismatic mini-fauna. And one of the reasons we study these mice is not only are they easy to work with in the field, relatively speaking, but also because they've been the subject of study by ecologists and natural historians from the beginning of the 20th century. So there's a huge wealth of natural history information that we can draw upon. Today, I'm going to focus on a study done by Osgood in 1909, who was primarily interested in describing this variation within the single species of mouse taxonomically. So he drove around the country over many, many years collecting mice. And one of the main observations he made was that he argued that there were two ecotypes, what he referred to as a forest ecotype and a prairie ecotype. And he argued that these mice were very easy to distinguish. In other words, if you handed him a mouse, he could just look at it and determine whether it's a forest form or a prairie form. And he argued this was the case because the two mice differed in so many traits. For example, forest mice tended to have darker pigment, longer whiskers, bigger ears, longer feet, longer tails, and they also preferred to climb and did better climbing. What's even more interesting is that many of these traits probably arose quite recently, because it's likely these forest forms evolved from a prairie ancestor only about 10,000 years ago, when the glaciers retreated after the last glacial period, opening up forest habitat. And we've now confirmed that some of these forest prairie populations are indeed estimated to be about 10,000 years old using molecular data. Finally, we think that these mice forest forms evolved from the prairie ancestor not once, but multiple times independently. So again, using molecular data, we've shown that there's been independent evolution of forest forms both on the east and west coast of North America. And what this means is that we have two independent evolutionary replicates. occurring in the east and west of the US. This suggests two things to us. One, it supports the idea that many of these traits may in fact be adaptations to forest habitat because they've evolved repeatedly, at least twice on the east and west coast. And second, it allows us to ask some interesting questions about both the proximate and ultimate mechanisms driving these differences, and if they're the same on both in both of these replicates. So what this scenario then led us to do was to go out in the field and collect mice from the east and west coast of North America. So one expedition, we headed out to Oregon, collected mice from the coastal forests of Oregon that represent the western forest ecotype, and then the prairie form from eastern Oregon. And then we also have an eastern forest and eastern prairie form from the East Coast, one from Michigan and the other from Pennsylvania. Now, the first thing I want to mention is that for the rest of the talk, I'm going to color code forest and prairie mice, forest mice in green and prairie mice in brown or tan. And so in these expeditions, we brought these mice into the lab, and we did this for several reasons. One, we could breed these mice. Two, we could get developmental time series. And three, we could ask the simple questions about heritability. So we brought these mice into the lab, and the first thing we wanted to do was to ask, do they in fact fit Osgood's pattern? Do forest mice have longer tails than prairie mice? And this was a particularly important question to address in the lab where we could control for environmental conditions, because we know from laboratory mice that the temperature at which you raise mice can affect tail length. So when we did that, lo and behold, what we found was that forest mice indeed had longer tails, both in the fields and when they were raised in the lab and across generations in the lab. So the first thing to note is that forest mice, again shown in green, have tails that are longer than the length of their body. So here you're looking at the on the vertical axis, tail to body length ratio. So forest mice indeed have longer tails than their geographically proximate prairie counterparts. The second thing to note is that the difference in tail length is approximately the same on the West Coast and the East Coast populations. And then the third thing to note, which we don't really have an explanation for, but is worth keeping in mind, is that the West Coast mice have longer tails than the East Coast mice, even within a habitat type. So the West Coast mice, forest mice, have longer tails than the East Coast forest mice. And we're not sure why that is. Okay, so now we have these mice in the lab, and they have heritable differences in their tail length. So now that we sort of established this system and these two evolutionary replicates.
Can I ask a question about that point?
Yep.
One of the things that the grants did that I thought was a lot of fun was to plot the beak sizes of offspring and parents. And, you know, there was a little confusion, at least possible confusion, about who the male father was, but they thought they in general got it right. So, you know, you just used the word heritable. That makes total sense. I'm just curious, like, is there a corresponding thing that you've done where you actually correlate what's going on with parents and offspring?
Certainly, that's one way. That correlation between parents and offspring is one way to estimate at least one component of a heritability. We could certainly do this in a much more complicated way, or I would say robust way, by looking across generation, measuring tails, etc. We haven't done that because it turns out that at least that's to get a really good estimate of heritability. It turns out it's a lot of work. So for us, at least initially, it was enough to say it looks heritable, in the sense that the morphological differences that we see in the field are maintained in the lab in a constant environment, and that it continues across generations. And then I'll show you some more data about the genetic basis, which also is very robust sort of evidence that it's heritable if we can track down some of the genes. But I would say you could certainly do that, but it's a lot of work to get a really robust estimate of heritability, and that wasn't the main focus. But it's something that we actually could, in some sense, do because we have a lot of that data already. Okay. So now that we have these two evolutionary replicates, they're in the lab and we have a hint, although not robust measures of heritability. The first question we wanted to know was, why does tail length vary? So I can imagine you're already thinking that tails might be important for forest mice because they can be used in balance when the mice are climbing. And this idea is not a new idea. It was an idea that has been brought up, that Osgood raised, but was first tested by Elizabeth Horner when she was a faculty at Smith College in the 1950s. So let me describe the experiment that she did. So she brought Peromyscus into the lab, a forest mouse shown in green and a prairie mouse here shown in brown. And she had these mice walk across a horizontal rod. And then she simply measured the number of times the mouse fell off the rod. So on our vertical axis, we have the number of errors. So the higher up on that axis, the more times the mouse fell off the rod and the worse the overall performance. So the first thing to note is that not unexpectedly, at least based on this hypothesis, prairie mice did worse in this particular climbing assay compared to forest mice. Okay, now here's where the experiment gets slightly gruesome. What she wanted to know was, what is the contribution of the tail to these differences in climbing? So what she did was she chopped off the tails of both prairie and forest mice and then had them run the assay again. Now the expectation is that when the mice go back into this assay, even a few weeks later, having already sort of practiced on the rod, both mice would perform, both forms of mice would perform slightly better. So that the data points would go down. But what she found, in fact, was when she lopped off their tails, with the prairie mice, the performance was roughly the same, but she saw a considerable decrease in climbing performance in the forest mice. So what this told us was that tails matter for climbing, at least in this particular assay. And second, that tails matter more for forest mice than for prairie mice because their performance was more adversely affected than the prairie forms.
Hopi.
Yep.
One question is there any difference in body size between these two mice?
These two forms, because they're within a single species, I don't know about Elizabeth Horner's data. They're not big differences in body size, but I'll show you some data later that body size does matter for performance and that smaller mice tend to do better. In the data that I'm going to show you next, we controlled for body size. These data I'm not sure about, but they're not grossly different because they're potentially only separated by a few thousand years.
Sounds cool. And then one other question we got is there any correlation between tail length and the types of trees these guys like to climb or the locations? I mean, I'm assuming that requires a higher resolution field expedition maybe, but.
Yeah, so we've collected some anecdotal ecological data. So the first thing to say is that at least where we collected our mice, the prairie mice, there are no trees. And so they do climb in shrubs and so forth. So at a gross level, there's a big difference because forest mice do climb and prairie mice don't, but because of the available habitat. We haven't looked at a more micro scale. So for example, in populations in which they have a greater variation in tail length, if there's some association between, like, I guess one way to think about this is, does the mouse know how long its tail is and does that affect its sort of preference for climbing?
Yeah.
And that could be based on, some innate knowledge of their tail length or based on experience. Like, I have a short tail, I'm not a good climber, I fall off all the time, so I'm going to climb less. But we don't know.
Got it. Thank you.
Great question. And we have measured climbing in the wild. So we've taken wild mice and put them in a climbing assay. I'm not going to talk about that today, but there's some interesting data that are starting to emerge from that. Okay, so so Elizabeth Horner's experiment clearly showed that having a tail or not having a tail mattered. But what we were interested in was taking the same kind of approach, but in a more biologically relevant way. And that is, we use the same sort of climbing assay, but to ask, not does having a tail or not having a tail matter, but do these differences in natural tail length matter for climbing? And we wanted to do this in a much more sort of quantitative, automated way, as opposed to just counting how many times the mice fell off. So what we did is we built an arena with two raised platforms connected by a thin horizontal rod. we introduced naive mice that have never climbed in this assay before into the climbing box. They were put on one of the platforms and allowed to freely cross the rod for 5 minutes. And then we filmed this assay from 2 angles, from below and from the side, where we could start to automatically, or in an automated way, track the movement of these mice. And while this assay, of course, doesn't capture all the complexity of a natural environment, we do think it captures some ecologically relevant behaviors, because mice do in the field traverse these horizontal thin branches. Okay, so I'm going to show you some data from this assay. But first, I just want to give you a sense of what the behavior in the assay looks like by showing you a video of an eastern forest mouse and an eastern prairie mouse. Now, the first thing I want to say is that these are not extremes. This behavior represents the average sort of performance of a forest mouse and a prairie mouse. And so we'll look at the eastern forest mouse first, and what you can see is the 2 platforms, the thin rod, and this mouse has moved from the platform freely onto the rod. And you can see, we watch its tail, it uses it to both wrap around the rod and as a sort of stiff counterweight. And then we've devised a way to sort of automate the behavior using both custom and freely available approaches. Okay, so the eastern forest mouse, as you saw, did quite well crossing the rod. And this is in stark contrast to our eastern prairie mouse, which you will see here. Goes on the rod, but right away starts flailing, still using its tail to wrap around the rod to try to right itself. And then we can also automate the analysis of this poor climbing behavior as well. So these 2 videos sort of capture the behavioral differences that we can quantitate here. So as the video suggests, forest mice perform better than prairie mice in rod crossing. This is true for both the East and West Coast lineages. So one of the first things we decided to capture was the fraction of time the mice spent upright. As you can see in the video, that differed between the forest and prairie forms. The forest mice shown in green spend about 85 to 90% of their time upright, whereas the poor prairie mice, either on the West or East Coast, on average spend more time upside down than they do upright. And this is for 2 reasons. First, when a mouse is upright, the duration it stays upright is longer in the forest mice compared to the prairie mice. And second, when a mouse falls upside down, forest mice are able to right themselves more quickly, so the bout duration for a upside down forest mouse is shorter than a prairie mouse that takes a longer time to regain its upright posture. And then the third thing we looked at was the speed at which a mouse, when upright, can traverse the rod. And what we found, again, as predicted, is the forest mice are able to move more quickly than the prairie mice. Okay. So I think what these data suggest is that there's a correlation between tail length and performance, and hint at potential ultimate mechanisms that are driving these differences in tail length. And this is a point I'm going to return to later. So now that we have this kind of hint at why tails might vary, the next question I want to ask is how tails vary. So now diving into the proximate mechanism. And we're going to address this question at 3 levels. First, we're going to start at the level of morphology, about how tails get longer at a morphological level. So if you think about a tail is comprised of caudal vertebrae, which are just segments along the tail. And if you're going to increase the length of a tail, in the most simple way, we can increase the length of that tail in 2 kind of fundamentally different ways. One, we can keep the number of segments the same, but increase the length of those individual segments. On the other end of the spectrum, we could keep the length of the segments the same, but just add segments. And in both of those, using both of those sort of approaches, you could achieve a longer tail. So we're interested in how did forest mice on both the East and West Coast get longer tails, and to address that question, we took X-rays of the tails and then came up with an automated way to measure the length of the tail, the length of the longest vertebrae, which is correlated with length of other vertebrae, and count the number of vertebrae. And then start to ask, how do they
Wait,
get longer
I've
tails?
got a question for you.
Mhm.
But before you knew the answer, did you have a guess? And the reason I'm asking is that you probably know about this work from Andy Oates, which has to do with mutants in some mitogenesis, actually, in zebrafish. And so there, in that case, the actual number of somites changes. And so, yeah, I'm curious, if going in, well, maybe you already knew the answer, kind of, or whatever, because I'm not sure offhand which way I'm going to go.
Yep.
So, yeah.
Interestingly, so in the mouse literature, there are lots of tail length mutants in laboratory mice. There are over 150 where the mutation results in a shorter tail, and only 2 and maybe 3 that result in a longer tail. So based on at least the mouse literature, we didn't have a good guess because we just didn't have, there's not a lot of tail lengthening mutations. And that's what the direction we thought that evolution took, went from sort of longer, from shorter to longer. So I think we went in pretty agnostic. And I think in the end, that turned out to be the right answer. And I'll show you why in a second. Okay. So having no preconceived sort of thoughts about which of the two solutions that evolution used, we measured both. And what we found is the answer was evolution sort of used both, in that forest mice have both longer and more vertebrae. And so here what we're looking at is on the vertical axis, longest vertebrae, and on the horizontal axis, sacrum length, which is a measure for body size. So what you can see for every body size, the longest vertebrae is shorter for the prairie forms compared to the forest forms. And when we count vertebrae, what you can see is that there are more vertebrae, on average 28, let's say in the western forest mice, compared to 24 in the western prairie mice. And so, in other words, evolution has sort of taken advantage of both of these approaches. Oh, and one thing to answer Rob's question, there was data in which folks have artificially selected for longer tails in laboratory populations of mice. And what they found, if I'm remembering correctly, is that you could get longer tails either by getting longer vertebrae or getting more vertebrae. But they never found a case in which you got both.
We have a question that's kind of related to all this,
Yeah.
which is the question of within a group. In other words, if you look at just forest mice,
yep,
and you note the variations within, does the behavior go along for the ride or not? In other words, if you have a longer tail and you're a forest mouse, are you better? Are you the Michael Jordan of your cohort?
Yeah, that's a great question. So we have that data because we have, for each individual, we have tail length and vertebrae number and vertebrae length and the behavior. We haven't analyzed that within a ecotype yet, in part because we have much less power because there's much less variation. But I think that's a really interesting question that certainly we'll get to, but I don't know yet. We started with the easier between ecotype comparisons first.
And another question related to this discussion is it common for mammals, in mammals, for the number of vertebrae to vary within that species?
So most of the studies that have been done have been across species, and much less within species. Obviously, within a species, there's just tends to be one less time for divergence to occur, and because there's tends to be homogenizing gene flow. So one of the things that was exciting about Peromyscus maniculatus is that it's such a widespread species and occurs in these dramatically different habitat types. It's sort of the best case scenario for intra-specific variation. But I can't argue that this is the case where there's the most variation. Oops, I have a message from my son. He needs my phone. Classic. Thank you. Okay. Okay. Here we go. So, are we good with questions? Good.
Yeah, I think we're good for now. There are other questions I we'll ask at the end. So
Okay,
just to continue with the thread that you're on right now.
Okay. So they use both. And what's even more interesting is that these two sort of solutions contribute equally to the difference in tail length. So here we're looking at cumulative tail length on the vertical and moving down the tail on the horizontal. So here's the prairie and here's the forest on the West Coast. And what we estimated is about 50% of the difference between the prairie and forest is due to the number of vertebrae. And 50% of the difference is due to the length of those vertebrae. And what's even more remarkable is that it's the exact same ratio on the independently evolved East Coast. So why would you have such similar morphological differences that both length and number matter and matter to the same extent in both evolutionary lineages? So one idea is that there's a functional explanation. So if you remember back to the video, the mice sort of used their tails in two ways. One, they would wrap their tail around the rod when they were slipping. And the second, when they were moving across the top of the rod, they would hold their tail out stiffly and use it sort of as a counterbalance as they moved across the rod. So it could be that having many vertebrae maintains the flexibility to wrap, but not having too many, so that you know, having longer vertebrae may help you keep your tail stiff. So it could be that this combination is important to maintain both of those functions. But the other reason you could have these equal proportions is because, or use both of these approaches, is because of genetic constraint or pleiotropy. In other words, mutations that affect the number of vertebrae could also affect the length of vertebrae. In other words, you can't disentangle those two. Well, it turns out that this is a relatively straightforward question to address using genetic crosses. So what we were able to do with our lab populations is take short-tailed mice, which have brown chromosomes, and long-tailed mice, which have green chromosomes, cross them together, generate F1 hybrids that have, I should note, intermediate length tails, then cross these together to get this large genetically heterogeneous population, each individual having its own unique tail length and its own unique combination of alleles. And we did this both for the cross on the West Coast and independently for mice on the East Coast. And simply ask the question, in these genetically heterogeneous mixed up hybrids, could you dissociate the length of vertebrae and the number of vertebrae? And when we did that, what you can see here is vertebrae number on the horizontal, the length of vertebrae on the, on the horizontal, vertebrae number on the vertical. And you can see this looks like a shotgun pattern, and there's no correlation between number and length. Similarly, we see the same pattern on the East Coast. So what this means, that lack of correlation in these F2 hybrids, is that in fact we can genetically separate number and length, suggesting that there are independent mutations that affect those two traits. Okay, so this leads us to think that maybe there's a functional mechanism, a functional reason a functional reason for having both differences in length and number. Because there are two different genetic mechanisms, this led us naturally into the next how question of what are those genetic mechanisms?
Hey,
So
Hopi, can I ask you two questions before you get to this next part? One is a little bit, corresponds to a few slides ago, but it's, I think, relevant. Is during development, when do tail differences emerge? And, this might be related to what you're going to talk next. Yep,
we'll talk more about that later, but the quick answer is that segmentation, the number of vertebrae, is an embryonic process, and the length of vertebrae seems to be mostly postnatal. So developmentally, they occur at different times, although there are mutations known to affect both traits.
Okay, so that already gave you maybe a clue that they couldn't be pleiotropic because they occur at different time points. Is that what you're saying?
I think in retrospect, yep, that's how I would interpret that. Although I will admit that we didn't think very much about that initially, and maybe that was naive, or there isn't that much data on it.
Yeah. And then another question going back to the videos is, in your videos, the prairie mouse was still able to wrap it still around the rod. So what's the behavioral advantage of having a long tail? Is it counterbalance, extra paw, you know? So
I would say at this point, we don't know precisely. But one of the things that I'll mention at the very end, one of our sort of next steps is that the videos that I showed you and the sort of automated behavioral analysis, which is much more sophisticated now than what I showed you in those videos. But initially we focused just on watching where the mouse was, right? Like, was it under the rod? Was it on top of the rod? Did it fall off, etc. But now what we've been able to do is to track all sorts of things like paw position. But most relevant to the question is we've been tracking the tail. So this is work by my graduate student, Emily Hager, and she's just finishing analyzing these data, asking, do the prairie and forest mice use their tails differently? And how does that affect performance on the rod? So we don't really know the answer in any sort of quantitative way yet, but we have the data to answer that question.
Thanks.
That's really fun to find out. Okay, so we made these big crosses, and for all of those mice, we measured length and number of vertebrae, which is the data I showed you just now. But for those same mice, what we can do is genotype them at hundreds of thousands of markers across the genome and ask, are there genomic regions in which genotype correlates with some aspect of morphological variation? And that's our forward genetic approach, also known as QTL mapping, quantitative trait locus mapping. And what that allows us to do is to localize in the genome regions of the genome in which there's a statistical correlation between genotype and the trait of interest. So we started first by looking just at total tail length. And here's our statistical correlation, our LOD score between genotype and phenotype. And there were 6 regions across the genome, which I'm highlighting here with their chromosome numbers, that showed an association between the length of the tail and genotypic or genetic variation. Okay, and this is not surprising in the sense that this didn't look like a single Mendelian trait. It looked much more sort of continuous. So there seemed to be 6 regions. But then we could ask another question. We could look not just at total tail length, but instead at its component parts. So we next asked, what about the length of the longest vertebrae? And when you do that, you see those same 3 QTL regions implicated that are that happen to be on the right. So we see a signal on chromosome 17, 20, and the X that contribute to the length of vertebrae.
Hey, Hopi, do you mind if I interrupt you? I mean, you've used this to great advantage so many times, and I've definitely read about it quite a few times, but can you just give a thumbnail sketch of, you know, kind of what happens? Because I think I understand what you're concluding, roughly speaking, like you're finding targets on the genome, basically. But what I'm not clear on is what's the thing that leads to a plot like this?
Okay, so nope, that's great. So here, let's say we're looking, can you see my cursor?
Yes.
Okay, good. Okay, so here's chromosome 3. Each one of these little dashes represents a polymorphic variant this is in that big F2 generation. So these are the mice that have different tail lengths and have different genotypes. Their genotypes are all mixed up between prairie and forest forms. We genotype them and try to find variants that in the parents are fixed. So the forest has an A and the prairie has a T. And then we ask what each of these F2 hybrids has at, again, hundreds of thousands of markers to the genome. And when you see, I know I can't remember which direction I went, but if the forest allele is associated with long tails in the F2 hybrids, and so every time an F2 hybrid has a forest allele, it has a slightly longer tail, you're going to start to see that correlation between genotype and phenotype. And that's where this LOD score, which is that measure, starts to pass over this genome-wide significance threshold. And that suggests that somewhere in here, there's a gene or genes that contributes to that difference in tail length. So these genetic markers are just looking for those correlations, and it suggests that somewhere in that region, there's a causal mutation.
So then let me ask you a little bit of an elaboration. Sorry, maybe this isn't fair to everybody else, and you and I should do it later. But like in the case of coat color, you found mutations, and we're talking single point mutations in the melanocortin-1 receptor. And so what I don't understand now is what's the deal with the huge length scale here when something might be a single point? Like, why doesn't it show up as a delta function more as opposed to like some blob? So, and then I'll stop talking to you about it.
Okay, no, that's a great question, and that sets up the big challenge too. So the reason that you don't see just a single peak or very narrow confidence intervals, which is what these color codes are showing you, the confidence intervals. And in some of these, there are hundreds, if not thousands, of genes that are correlated, and that's because we haven't had enough recombination. So we just did one round of recombination. The frequency of recombination in the mammal genome is not that much, and so you get these big blocks. And this is why if you do a GWAS study, for example, in a population, let's say we had a population where we had long tails and short tails that have been recombining in the wild for thousands of years, the recombination has occurred so much that that block gets broken up, and you can really narrow in on the causal mutation. So there's a trade-off. Here, the sort of signal is bigger, so the signal's easier to find. But then you're left with this big chunk to try to find the causal mutation. Whereas if you do a GWAS, you might, if you find something, you're closer to the causal mutation, but it's harder to find. Okay, so that brings up the big challenges that we'll try to address shortly, and that is, how do you go from a big QTL region to a causal set of candidate genes to ultimately the causal mutation? And that's where we spend the vast majority of our research time, is tackling that question. For pigment, we've been really lucky because we could then pick a region of the genome and say, okay, of these 100 genes, have any of them previously been implicated in pigmentation phenotypes? And often there was a gene like the melanocortin-1 receptor or a GUDI or these obvious candidate pigmentation genes, and we were just fairly lucky that we could then, functionally verify them. And I'll tell you a little bit more about how we can do that with these skeletal traits. But it becomes much harder when we're talking about behavioral variation, because we don't even know what a good candidate behavior gene would look like. Okay, so back to this sort of bigger picture. So we have these 6 regions that have been implicated in total tail length. 3 of them seem to be associated with the length of the longest vertebrae, and the 3 other ones seem to affect the number of vertebrae. And I love this result in part because it's so beautifully symmetrical, but it sort of makes 3 important points. First, this explains exactly why we could genetically dissociate length and number, is because those causal mutations are on different chromosomes. Second, what we found is that of the variation that occurs between the prairie and forest form, about half of that variation can be explained by these 3 QTL, and half of the variation by the 3 purple QTL. And that's consistent with both of these traits contributing equally to the difference in tail length. And then the third thing I'll tell you is that in each of these 6 cases, it's the forest allele that's associated with the longer tail as opposed to the prairie allele. And because that's true for all 6 QTL, that's consistent with selection driving these differences in tail length. Okay. So now we get to Rob's question of how do you go from these big chromosomal regions to causal genes? So what we decided to do was first focus. Oh, I also wanted to tell you before I get to that, the data I'm showing you here is just from the East Coast cross. We did the same cross for the West Coast mice. So we could ask, are the same genetic regions, chromosomal regions, implicated in variation in the difference in tail length in the West Coast? We found QTL on the West Coast. They're shown here, 5 of them. But when we line them up with the QTL from the East Coast, there's almost no overlap, with maybe one exception, although it's unclear if this is the same region or not, which suggests that there are completely different genetic regions contributing to long tails in the East and West Coast. And this to us was a really big surprise, because again, what I showed you earlier is that morphologically, they're very similar, right? They both have more and longer and in the same ratio of vertebrae. One thing I will note is that if you see here on the West Coast mice, we have this really interesting peak on chromosome 15. It turns out this is a big inversion, and that inversion is associated with longer tails. And we've been working on a fun story on that population. But for now, I'm just going to focus on the East Coast and return to this. So here we're getting to Rob's question now. So when we were thinking about genes within these QTL regions, we decided to initially just focus on the number of vertebrae, in part because this is an embryonic phenotype and hence slightly more tractable. And we decided to focus on one QTL peak, which was the biggest peak shown here on chromosome 8. And because it happened that this peak included some pretty exciting candidate genes. Okay, so how do we go about finding these candidate genes? So Rob, this is the big question. So in this particular QTL interval, there were 140 protein coding genes. So what we asked is, of these 140 genes, which ones showed differential expression in a developing tail bud? And so we dissected out the tail bud at biologically appropriate times during embryonic development. We found thousands of genes that were differentially expressed, about 3,000 genes. But then we could ask, of those 140 genes, how many show differential expression? Or in other words, of those genes that were differentially expressed, how many of those were in the QTL region of interest? And it turns out there are 25. Okay, so 25 is a manageable number. But then we took advantage of the fact that we do know about genes that affect tail phenotypes in laboratory mice. And of these 25 genes, 3 of them are known to have phenotypes. 1 second. This is classic Zoom.
It's a friendly version of bombing.
Okay. So now we're down to three genes that at least are at the top of our candidate gene list. Okay. And those 3 genes, 2 of them are Hox genes, and one, SP5, is involved in Wnt signaling and important for embryonic patterning. So of these 3 genes, one in particular caught our attention, and that's this one, HOXD13. So for those of you who remember back to Developmental Biology 101, Hox genes are really interesting in part because of their role in patterning, and in particular skeletal patterning in vertebrates. And one of the things I always remember about Hox genes that was so fascinating was that they're collinear. In other words, the position of a gene in the Hox gene family mirrored where it was expressed in the embryo. So HOX1 genes were expressed in the head, whereas HOX, for example, 13 genes were expressed in the tail. So this was exciting because HOXD13 is expressed in the right place, that is in the developing tail bud. Moreover, we know from beautiful developmental biology studies from Olivier Porquier and Jacqueline Deschamps that if you turn HOX13 genes on in a developing mouse, that causes early axial termination, in other words, short tails. So we know that HOXD13 expression can affect tails. Now with this in mind, let's go back and return to the HOXD13 pattern. And what we see in this pattern during early tail development, the short tail form has high amount of HOXD13. So potentially a signal, let's stop growing our tail. Whereas it's low in the long-tailed forest mice. But late in development, that's where we see an increase in HOXD13 expression in the forest mice, potentially saying, okay, forest mice, now that we have a long tail, we can turn off tail development now.
Can I ask a little clarification point that came up in the Q&A?
Yep.
It's just the question of specifically, you probably said, but both this person and I might have missed it. How are you detecting differential expression? Is it on transcriptomics or what?
Yep, sorry, I skipped over a lot of that detail. So we did RNA-seq on tail buds that we dissected out at mouse time points 12.E, 12.5, and E15, I think. It's on a later slide. So early in sort of tail development and then later in tail development. So those 2 time points. And then we've combined those data sets, but we also looked at them independently. And we had very, for those who are sort of RNA-seq aficionados, we used very low threshold for differential expression because we thought that, especially during embryonic development, even small changes in expression could have large impact on morphology. So that's why we have so many differences, even though these, they're just two ecomorphs. So 3,000 was not surprising given our threshold was so low. Great. Sorry for that lack of detail.
Thanks.
Okay. So we got really excited about Hoekstra D13, and let me just set expectations. We have made transgenic animals, and we are analyzing them now. So we don't have the nail in the coffin experiment, but we thought if this is our leading candidate gene, not only did we want to functionally validate it, but we also wanted to understand how this gene or potentially another gene acts through development. So what's happening during development that gives rise to differences in tail length? And so here I'm going to show a lot of data in an abbreviated form, but just to provide some context, so tail development in laboratory mice occurs primarily from E10 to E15-ish. That's really when the tail is growing. And the somites, which give rise to the vertebrae, are formed. So this image down below is actually of a chick embryo in which a lot of beautiful work has been done on semitogenesis, especially in caudal vertebrae. But let me just sort of explain a simple way to think about how this works in vertebrae. So we have this material called the pre-Semitic mesoderm. This is kind of the stuff from which slices will be taken that form somites that form that ultimately it will develop into the caudal vertebrae. So we had this question about how do you get more somites? How do you get more vertebrae? And we came up with two sort of hypotheses. One is you could take that pre-Semitic mesoderm, let's think of it as a loaf of bread, and you could just slice it faster into thinner slices, faster/thinner slices, and then you could get more somites. The other way you could get more slices of bread would be if you start off with a loaf that's bigger. In other words, you have more pre-Semitic mesoderm. So we tested those two hypotheses, and what we found was, in fact, there are differences in the pre-Semitic mesoderm, and we don't find any evidence for the timing of the slicing of that material to be different. So here's the data. Here we're looking at the length of the pre-Semitic mesoderm. Here we're looking at a marker for time. And what we see is that we see a difference in the pre-Semitic mesoderm across development and in the direction we expect, and that is the long-tailed forest mice have more pre-Semitic mesoderm. In other words, their loaf of bread is bigger. Okay, so this was really exciting, but then of course we wanted to know, well, what gives rise to the pre-Semitic mesoderm? There are some hypotheses in the literature, and one of the things that we decided to look at was this, what are called neuromesodermal progenitor cells, which have been shown to be what gives rise to the pre-Semitic mesoderm. I'm skipping over a lot of detail here. But we decided to stain for these NMP cells in the chondroneural hinge using these markers. And what we found is that we see a difference in the number of these NMP cells, and again, in the direction we expect, with the forest mice that have the bigger pre-Semitic mesoderm having more of these progenitor cells. And what's particularly exciting, although very, very, I would say preliminary, is that there's a suggested role for Hoekstra D13, and I would say suggestive, emphasis on suggestive, in maintaining these NMP cells. So there suggests there's a potential link between Hoekstra D13, these NMP cells, which give rise to the pre-Semitic mesoderm, which ultimately give rise to the somites and the vertebrae. Okay. So let me just step back and summarize what I've told you so far. So we started with this natural history observation made in the early 1900s that there were these two ecomorphs within the single species of deer mouse. I showed you that when we brought these mice into the lab and we designed this climbing assay to look at performance, that we saw significant differences in climbing performance between forest and prairie mice, with forest mice doing significantly better in a number of measures. Then I showed you that one of the reasons may be this difference in tail morphology, with forest mice having longer tails, both because they have longer vertebrae, caudal vertebrae, and because they have more caudal vertebrae. I suggested one of the reasons they might have more vertebrae is because they have a larger sort of pre-Semitic mesoderm, the stuff from which the vertebrae emerged. And this may be in part, although this link is still quite tenuous, in part because of Hoekstra 13. Okay, so if you're gonna look at this story that I've told you so far, there's really one missing link. And so this is the last piece of data I want to show you. And that is
a max of about 5 minutes. Just
perfect.
In case.
Okay, I have 3 more slides. And that is that there's a correlation between tail morphology and climbing performance. But as you know, as I mentioned before, there's all sorts of other differences between forest and prairie mice, from longer whiskers to bigger feet, that also could contribute a role. So how do we show that tails actually matter for climbing? Well, we could return back to these recombinant mice, remember, that have all different lengths of tails, but are genetically sort of mixed up in all sorts of other traits. So this population of mice allowed us to dissociate tail length from, let's say, foot size and whisker length and other traits. And then we could assay them for climbing performance and their tail morphology. And in this population, ask, is there a correlation between your tail morphology and your climbing performance? And when we did that using a canonical correlation analysis, what we found is there's a significant correlation between morphology and performance. And what I'm showing you on each of these axes are the variables that contribute to those differences and the weight of those variables. So what this tells us is that having lower weight and a smaller mouse that weighs less tend to do better, but that mice that have longer feet and, importantly, longer tails do better. And by doing better, that means they spend more time upright, less time upside down, and when they're upright are faster. So these data really suggest that longer tails are important for climbing performance. Okay, so we have started to make some of these connections. So we've measured these differences in morphology. We've shown some evidence how these morphological differences can affect behavior. I've suggested at least a nice candidate gene for these differences in morphology and a potential mechanism by which this gene acts through development to cause those changes. Okay, but clearly this picture is not complete. So I'll just tell you very quickly the 2 directions we're taking. So our unanswered questions. The first one involves these linkages about how morphology and behavior both together and independently affect fitness. And in this case, I mean performance. So as I mentioned earlier, we can track mice, the mouse itself, but we've also been able to now track the tail, which ends up being a very difficult problem. But Emily, my wonderful grad student, has seemed to have solved it. So now we can ask questions like, how does vertebrae length and vertebrae number affect tail use? And do forest and prairie mice use their tails differently? We're also spending a significant amount of time trying to make these linkages, which I didn't touch on today. So I'm just going to tell you one cool piece of data that we discovered, and that is we were looking at the cortical spinal tract and comparing forest and prairie mice. And initially we were doing this just to check antibodies to see if they would work. But what we found is that the cortical spinal tract is significantly larger in forest mice compared to prairie mice. And we now are replicating this experiment. And so what we want to ask in this context is, what is the genetic basis for these? So here we see a difference in the circuitry. What is the genetic basis for variation in this neural anatomy? And what are the effects of these differences, if any, on behavior that gives rise to differences in performance? Okay, so that gives you a sense of where we're going. Let me end by just thanking, in particular, Evan Kingsley, who started this project as a graduate student. He's now a postdoc in Cliff Taben's lab, pursuing his interest in Evo-Devo. And then this project was taken up by Emily Hager, who did all the behavior work, who just finished last month and is starting up a postdoc at BU. We had a ton of undergraduates who helped us, a lot of people in the lab who contributed to this work, both in the lab and the field, and my funding sources, and I'm happy to answer any remaining questions. Thanks.
Thanks, Hopi. I say you unequivocally lived up to your introduction. I haven't heard.
It's
Really
impossible.
fun. Well, okay. Anyway, Aaron's gonna tell you some of the other questions. But thanks, that was fantastic. Is that written up yet?
We are just writing both the story of the East and the West Coast at the moment. So pre-submission inquiries are out.
Connect it to East and West Coast rappers while you're at it. Okay.
All right. Yeah, that was great, Hopi. So let's see. There are a few questions here that are, I think, relate to various topics throughout the talk. One that I thought was very interesting is how it relates to nature versus nurture. And so the question was there a control done for mice growing up climbing versus not growing up climbing? You know, how much is this a learned behavior? I guess that's.
Yep. So as a geneticist, I tend to like to ignore the learn component or control for the learn component, I would say. So at least in this assay, what we did is we took mice that all the data I showed you was from mice on their first trial, the first time that they went in the assay. We have looked at mice when they're given multiple opportunities to climb across the rod, and they do get better. And it's a little bit hard to disentangle how much of that is them being less anxious in this new behavioral arena versus actually learning to climb better. And we haven't tried to disentangle those two. And we also haven't done the experiment yet of giving them the opportunity to, let's say, climb on other things. One caveat to note is that what I said is these mice haven't climbed in our behavioral assay, but they can climb in their cage a little bit, and and maybe forest mice climb on their cage a little bit more and therefore have more practice to begin with. And we haven't really controlled for that, but I can tell you that in this assay, they're both naive to the assay. And the last little piece of data is, as I mentioned, we took this climbing assay out into the field and had wild mice climb in there, and we're just analyzing that data now. But I suspect we're going to see big differences between forest and prairie mice. And those differences may be bigger because forest mice have been climbing their whole life and prairie mice have at least only been climbing in shrubs. And so they may be more exaggerated, but we'll see.
Mm-hmm. And is their lifestyle such that the little ones learn from the adults, or not really?
Hard to know. Okay. They could see their mom climbing and they climb, or their mom climbs more, so they climb more. But I don't know.
Okay, so so one question about the F2 recombinants and the QTL mapping, I guess, is, you know, the F2 recombinants could also have differences in all the phenotypes, right? Other than the tail. Like, okay. All right, but I guess you guys don't score for that, or how does that work in the context of QTLs?
Yeah, so whenever we do a QTL, I'll admit we sort of measure everything we can, because you just get, you have potential for more hits. So we have measured foot size, because that's also a trait that Osgood noted that could have an effect on climbing performance. And there's very weak correlation between tail length and foot size, suggesting most of the genetic mechanisms are independent. And that's why doing that last experiment I showed you, where we took those F2s and we put them in the climbing assay, then we get mice with long tails that have big feet and mice that have long tails that have small feet, and allow us to sort of disentangle the effects of morphological differences on performance. But we do have QTLs for foot size. And in another project, we're looking at, we have wonderful QTLs for whisker length. And we know whisker length has big impact on climbing as well. Though none of those overlap with tail QTLs.
I see.
But I could give a whole talk on whiskers.
Say it again?
I could give a whole nother talk on whiskers.
So another question related to these crosses is, did you guys try to cross forest mice from both sides of the country?
Yeah.
Do they get longer tails?
Yeah, so that's the classic complementation test in genetics that works if all your mutations are recessive, which most of ours look to be semi-dominant, which is not unexpected for natural populations. We haven't done that experiment, but because we got the same data doing it independently in both populations about sort of the commonality of the genetic mechanism, I think it would be fun to do just to see how well those two data sets comport. But we didn't do that initially, and we didn't do it initially because we suspected that not all the long tails alleles would be recessive, right?
Mhm. Got it.
I love that question.
So one more, it's more related to the, I guess, the developmental aspect, but is the longer size and length of tail vertebrae compensated for? I mean, does it cause other cranial lumbar vertebrae to be smaller and shorter in the forest mice?
We looked at other vertebrae, primarily focusing on the sacral vertebrae, and those appear to be the same between forest and prairie mice. So there don't seem to be any differences except in the caudal vertebrae. So that suggests not only is there not like a trade-off between vertebrae, but also it's not that it's caudal vertebrae and sacral vertebrae that are changing in size and number in a positive correlation either. It's a great question.
Got it.
All right. Maybe I'll chime in with a couple of others. So one question that was asked was about what the fitness advantages associated with climbing. So this is something also that your former colleague now, I guess, Jonathan Losos, has thought about in the context of his lizards in the Caribbean. But yeah, so what's up with that? Maybe just a quick thought on that.
Yeah, so this question of, why traits change and understanding the sort of fitness of, so what I told you about, I was sort of calling it fitness, but really it's, I would say, one level below. We're really looking at performance, right? So that's one thing to say, to admit to, I guess. And measuring fitness is really challenging. So the way that we've done it with pigmentation is to build enclosures that have dark and light soil backgrounds, put mice in of different colors, and then sort of see who survives. So you could do something similar, sort of have a forest enclosure and a non-forest. You can see how that would be even more complicated than the experiment we did with pigment. But that would be one way to get at sort of fitness. There's lots of ecological differences between the two. There's some suggestion that forest mice nest up in the trees. They certainly use trees to evade predators. So this is just an anecdote, but when we catch mice in our transect, let them out of the cage, forest mice run straight to the nearest tree and go straight up it, right? That's so clearly that's a way to avoid predators, at least some terrestrial predators. But these are all sort of anecdotal stories. Getting at the actual, doing the experiment is much harder.
Gotcha. This is this mainly just a pandemic inspired question, but I think makes sense. So we've been already in my group, we think a lot about viruses, but people have been interested in zoonoses. And so the question from Manuel in my group was, he was curious whether you guys ever get nervous about handling these animals and possible disease and that kind of stuff. Again, it's not related to your talk, but perhaps just a cultural thing. All of us to hear a little bit about what it means to be a field biologist.
Yep. So Peromyscus are best known, and probably most papers have been written on them because they are vectors for hantavirus. Hantavirus, or it's known as the four corners virus. Yeah. And so in the field, we wear protective gear, but like a lot of respiratory viruses, being outdoors is the best thing you can do. So we have N95 masks that we wear in the field when we work with these mice. And there have been a few cases. This is when people go to cabins and they sweep up the feces and urine and inhale that, that's the primary mode by which you get hantavirus. And there certainly have been deaths associated with that, although few that have been linked to field biologists. And then the second thing I'll say that's highly relevant to the pandemic is that there was just a preprint that came out a week or two ago that suggests that Peromyscus can get COVID. And they have the mutation in the ACE2 receptor, and their ACE receptor that makes them likely to be carriers. And then they actually did the experiment. It's a little unclear again, in these experimental procedures, you really hit them hard and much larger sort of infections than you would see in the wild and how much they transmit to humans. But we're testing our mouse colony now to see if there's any evidence for COVID, which just to be precautious.
So two last questions. They're both kind of related. So do heavier mice need longer tails to balance? And does only the tail length matter, or how the tail length is achieved, meaning more somites or larger vertebrae.
Yeah.
So just maybe just a quick word, because we're almost out of time, but just a quick impression of those two.
So that second question I think is super fascinating, and we're definitely looking at that. So when we get that tail use data by tracking the tail, we'll be able to correlate that with number and length, and to sort of test this functional hypothesis that that having a balance between those two components of tail length is what's important. And so you have to stay tuned for that. And body weight, that's a great question. And we have all that data. And again, we'll get this out of the tail tracking to see if mice of different body weights use their tails differently and compensate in some way. So stay tuned.
All right, great. Thanks so much. That was really fantastic. So just for those of you that are left,
Thank you, Hopi.
Tuesday, we have Amy Gladfelter giving a talk, and that should be great. Sorry about the mix-up earlier in the week. Otherwise, I hope people have a great weekend and that you're healthy and happy. And again, Hopi, thanks so much. I'm going to send you a mail to schedule a Saturday, the first Saturday of the course next summer. Hopefully, you'll say yes.
Of course. Thanks for having me. This was great.
All right, nice seeing you. Take care.
Thanks again, Hopi. Bye.
Bye, guys.